So this experiment was performed by the scientist Griffith, okay, and in 1928, this scientist performed this experiment, and he discovered the transformation occurs into the bacteria. The characteristic of the rough cell as well as the pathogenic cell, that is pathogenicity, it is get transferred to the rough cells from the smooth cells, and because of that this transformation occurs. Then later the experiment performed by the scientists Avery, Macleod and Carty, they discovered that this transforming principle was nothing but the DNA, because they used the three enzymes at that time. And to prove this, these scientists used the three kinds of the enzymes. You know, the DNases enzyme means these are the nucleases enzyme and degrades the DNA molecule selectively. Then RNases enzyme, these are also the nucleases, they degrades the RNA molecule selectively. As well as third kind of the enzyme they took, that is the proteases, and these proteases enzyme degrades the proteins. So in one reaction mixture, DNA was degraded, uh, and there was no transformation occurred. There was no presence of the smooth cells into the media, um, as well as they also perform. So here you can see there is a complete absence of the smooth cells. But into the second reaction mixture, where they took the RNases as the enzyme. So what will happen in this extract? This extract, it is taken from the type third smooth cells. This extract will present all three kinds of the molecules, then nucleic acids, specifically DNA, RNA, and these RNases will attack onto the RNA and will degrade the RNA molecule. Hence, here intact molecule will be DNA and proteins. Okay, and then this in presence of the DNA and protein, these rough cells, type second rough cells are get converted into the type third smooth cells, and they obtain the colonies of the smooth. Here in this third reaction mixture, they took the enzyme proteases. This proteases enzyme degrades the proteins, and in this mixture, the DNA as well as RNA is remains in the intact form. And when this DNA and RNA intact form mixed with the live type R second cells, then when these cells are plated, then there is a conversion of the rough cells to the smooth cell, because here also DNA is in. But here, if the DNA is intact as well as the proteins are intact. So because of that, where the DNA is intact, their transformation took place. Here DNA is not intact in the first reaction mixture, but proteins and RNA are intact. So that in this first reaction mixture, when this reaction mixture is mixed with the type second R cells, then there is no transformation of these rough cells into the smooth cells, even though the presence of RNA and proteins are into the intact. And because of that, there is no transformation, means that this suggests that RNA does not plays the role in the transformation. Proteins does not plays role into the transformation, and here uh, DNA, where the DNA is intact, here they got the smooth cells, and because of that, they concluded that DNA is responsible for transformation of the rough cell into the smooth cells, and these rough cells, non-pathogenic cells, are get converted into pathogenic cells. Hence, the transformation occurs. This transformation is carried out by DNA molecule. Means the DNA has the power to change the characteristics of the cells. Means the whatever the genetic information it is stored in the genome, that is the DNA, and they definitely concluded on the basis of this evidence that DNA acts as the genetic material. And similarly, DNA is responsible for transformation. Then later on, several scientists studied this transformation or studied several bacteria also, other bacteria also, and they found this transformation occurs